Hello, everybody. First of all, I want to introduce myself. I'm Leticia Masha. I work at molecular laboratory, virology laboratory from Uruguay, CENUR Litoral Norte, Universidad de la República. This work that I will present to you was done jointly with a genomic platform of Facultad de Ciencias, also the Universidad de la República from Uruguay. The work I will present you is entitled Coding Complete Genome Sequences of 2 Bovine Viral Diarrhea 1A isolated from Uruguay. These are the co-authors that participate in the work also. As introduction, briefly introduction, I could say that bovine diarrhea virus is a major worldwide pathogen causing significant economic losses to livestock sector and affects cattle production and reproduction. It causes acute infection, fetal infection, that could lead to diarrhea, respiratory disease, bleeding disorders, and acute death. Or it could lead to subclinical infection, fever, infertility, immunosuppression. On fetal infection, it could cause abortion and birth defects. Transient in utero infection, calf born with acute infection. Calf could born also as persistent infected animals that could death, have poor performance, or develop mucosal disease. Or instead, it could have normal appearance or immunosuppression. So BVD causes a wide range of symptoms, as we said. Taxonomy. BVDB belongs to the genus Pestivirus in the family Flaviviridae. There are 3 BVDB species recognized, actually. BVD1 that used to be named as BVD1, BVD2, and hobby-like Pestivirus. Recently, they were renamed as Pestivirus bovis, Pestivirus tauri, and Pestivirus brasiliensis by ICTB. BVDB genome has a single-stranded positive sense RNA genome about 12.3 kb. It has one open reading frame that encodes a polyprotein of almost 4,000 amino acid residues, flanked at both ends by untranslated regions. It codes for structural genes and non-structural genes. In Uruguay, BVDB, briefly, cattle population of Uruguay is around 12 million heads, including beef and dairy breeds. Uruguay is an agro-industrial country. Our economy hardly depends on cattle industry. So cattle industry exports represents around 10% of our country gross domestic product. BVDB seroprevalence at farm level is about 80%. It's high. And vaccination is not mandatory. A little bit of background of BVDB. We began to work on BVDB almost 10 years ago. And when we start working on BVDB, we start practically from zero. We only knows about seroprevalence at farm levels. So now 10 years later, we know that BBTB causes in Uruguay reproductive failures, abortions, and other associated disease affecting mucosal, respiratory, and enteric systems. We did molecular epidemiology studies, and now we know that BBD1 and BBD2 species are present in our herds, particularly subtype 1A and 1I and subtype 2B. Subtype 1A is the predominant one in our herds. And by bioinformatic studies, we determined that it has been present since 90s. This subtype, since then, spread and evolved in Uruguay, giving rise to local genetic lineages that are divergent from those circulating worldwide, or what is more, there are the BBDB1A from Uruguay are genetically distant from NADL vaccine strain. That's possibly or potentially could cause vaccination failure. So the main objective of this work was to obtain the coding complete sequences of 2 Uruguayan isolates, 1A, named as this, by Illumina sequencing technology. As materials and methods, we first had to isolate these strains from serum samples. They were animals that had BBTB symptoms. So we isolate cell culture on MDBK cells that are permissive to the virus with medium plus 10% of horse serum plus antibiotics. These plates were incubated at 37 degrees with 5% of CO2 for 3 days. We did 3 consecutive blind passages. And as BBTB is a non-cytopathic virus, we determine cell infection by an antigen capture ELISA. For sequencing, first of all, we release the viral particles from cell culture. And for that, we did freeze and thaw cycles to release viral particles. This RNA was isolated then from culture supernatant using this kit from CENUR research, and RNA has to be reverse transcribed using random hexamer primers using this enzyme from TERM. Then cDNA was purified, and then we construct the library using Nextera DNA Flex library kit from Illumina. And then libraries were sequenced on an Illumina Minisec platform. Then adapter quality trimming and filtering of raw data were performed using BBDuc of Geneus Prime program. As a result, first, we successfully isolate the 2 Uruguayan strains as the antigen capture ELISA were resulted positive. And coding complete sequence of the 2 samples were obtained by specific clean reads of BBTV of both samples or strains that were mapped through BBTB1A Oregon strain, that is the closest genome, were mapped to our clean reads. So we would construct the coding complete sequences of our strains using Genei's algorithm of Genei's prime program. In this table, we resume the sequencing metrics and the session numbers of our samples. Using Oregon strain as a reference, we also identify in our coding complete sequences an ORF and of almost 12,000 nucleotides and for almost 4,000 amino acid residues was identified on the forward strain of both coding complete sequences. As I already said, using Oregon strain as reference. Uruguay strain were compared each other, and we saw that they were highly similar among them. And genetically distant to another Bovine strain. This is in agreement with our previous studies with shorter genomic regions, such as 5' UTR, UTR, and MPRO, enzyme, and A2 glycoprotein. We did that on a previous work. So as conclusions, these two strains represent the first Uruguayan BBD1A isolates, also are the first coding complete sequences from Uruguay that are published. So we say that our results adds valuable information and are the stepping stone for further studies. Actually, these results are were the stepping stone for a project that we are sharing with another group for design a vaccine that we expect that could be suitable for protecting our hearts. That's all from this work. Thanks for listening. These are the references. Thanks to CENUR Litoral Norte, Universidad de la República, Molecular Biology Lab, Genética de Microorganismos, Genomic Platform del CENUR Litoral Norte y Universidad de la República. Thanks.